Bismillahirrahmanirrahim. Assalamu alaikum. I am Dr. Shagufta Riaz, and this is your first lecture of zoology from chapter 10, that is Kingdom Animalia. This is the introduction that what we will going to discuss in this chapter. We will discuss how animals evolved or developed as we can see the animals around us or in the wild. We will also study simplest members to the more advanced animals of the kingdom Animalia and different characteristics of these animals, which will include development of tissues and organs, their internal digestion, their body organization, which is also known as symmetry, and their internal body cavities. Here are few interesting facts about the kingdom Animalia. Kingdom Animalia includes 1.3 million species of animals, which means it constitutes around 75% of the total known species of the living organisms right now. Plus, this kingdom includes variety of animals, that is from a microscopic trichoplex. Trichoplex is about 1 millimeter in size, while the giant blue whale is, which is the scientific name of this giant blue whale is Balænoptera, and it has 40 meters length and about 1,60,000 kg in weight. These are important to learn for the MCQs. Kingdom Animalia is divided into 33 groups, which are known as phyla. The scientific term for these groups is phyla. Phyla is the plural and phylum is the singular. Out of these 33 groups, we have 9 major phyla, and these 9 major phyla will be discussed in this chapter that that is the Kingdom Animalia, while the rest of the phyla are minor phyla out of these 33 groups, right? And what are the major phyla that we will discuss in this chapter? The names are Porifera, Cnidaria, Platyhelminths, Nemathelminths, Annelida, Mollusca, Arthropoda, Echinodermata, and Chordata. To understand on what basis we classify animals, we have to know what is taxonomy or systematics. Taxonomy or systematics is simply the classification or grouping of animals on the basis of their evolutionary relationships, and these evolutionary relationships are found where? They are found in the development pattern of these animals, their cellular organization, and their diploblastic and triploblastic organization of their body, the symmetry, that is the overall shape of that animal, coelom, that is the body cavity. We will discuss all of this in detail in further slides, and pate of blastopore. So first, here we will discuss the developmental stages, the initial stages of any embryo, which include the development. As we all know, the animal life starts from a cell, that is zygote, which comes after the fertilization, and after the zygote is formed, the division starts. And first the mitotic division will start, and it will divide into two, then it will divide into 2 cells, and then into 4 cells, and after that it will divide into 8 cells, and then multiplication of the cells will go on, and a number of cells will form. The first stage will be morula, as you will find the detailed picture in your textbook. For here, just for the reference and just to show you the differences between these diagrams, I have taken this picture, right? After morula stage, there is a stage which is known as blastula. During development, after morula, blastula is formed. Blastula is basically a hollow ball, which means here you can see it is empty inside. The cells are formed on the outer side, right? When they multiply very quickly, they will form this like this a ball, form a ball like this, and which will be empty inside. This is a cross section of this ball. After further development, the next stage is gastrula, and in gastrula, these layers will differentiated and further layers are also forms. That is the three germinal layers, which are ectoderm, endoderm, and mesoderm. The outermost layer, this layer, the outermost layer is the ectoderm. The innermost layer, this yellow one, is endoderm, and this layer, that is here you can see this layer between the ectoderm and endoderm is mesoderm, right? And after the further development, there is an opening formed in this cellular structure. This opening is known as blastopore. Remember these three layers and the blastopore during gastrular stage, because it will be discussed in further slides in further topics. Now, another characteristic on which the animals are classified is their cellular organization. As you all have learned in your previous classes that the always different number of cells forms, and these cells together form the tissue, and then different tissues together form then organs, and different organs make an organ system, right? Which makes our organism a complete organism, right? On the basis of cellular organization, there are two further division of the kingdom Animalia, that is subkingdom Parazoa and subkingdom Eumetazoa. Subkingdom Parazoa include animals which lack a proper tissue organization, which means this subkingdom will include animals which have only cellular structure. They don't get developed into tissues. And the only example of the subkingdom Parazoa is phylum Porifera, which include sponges. We will discuss it in further lectures in very detail, right? Subkingdom Eumetazoa include animals which have tissues organized into organs in lower groups, and organs developed into united and develop into organ systems in the higher group of this subkingdom. And the example of this subkingdom Eumetazoa is the rest of the 8 phyla that we have discussed in the previous slide. The one is the phylum Porifera, which comes under the subkingdom Parazoa. Obviously, the rest of the 8 will come under the subkingdom Eumetazoa. The examples are here: Cnidaria, Platyhelminths again, Nemathelminths, Annelida, Mollusca, Arthropoda, Echinodermata, and Chordata. Remember these names. Another basis that on which the classification of animals depends is the diploblastic and triploblastic organization. To understand this, we have to see the introduction, in almost all eumetazoans during development, 3 tissue layers are formed, which I have shown you in the previous slide, the diagram. The ectoderm, endoderm, remember ectoderm, endoderm, and mesoderm, these 3 layers. The ectoderm was the outer layer, endoderm was the inner layer, and mesoderm was the middle layer. These layers are also known as germinal layers, as I told you before. So the animals which have developed only ectoderm and endoderm during their development, they are known as diploblastic animals. That means they don't have the mesoderm, right? And the example of diploblastic animal is only Cnidarians. Phylum Cnidaria comes under this diploblastic organization, right? In triploblastic animals, these animals successfully develop all 3 germinal layers, that is the ectoderm, endoderm, and mesoderm. So rest of the phyla except Porifera comes under triploblastic animals. Why? Because poriferans do not successfully develop these layers. That's why they don't come under the triploblastic animals. Symmetry. Symmetry is the overall shape of an animal, and on the basis of this we can call, there are two main types, that is symmetrical animals and asymmetrical animals. What are the symmetrical animals? That can be divided on at least one plane into two identical or similar halves, right? If we just take this definition into a different or into opposite one, that is the asymmetrical, always remember that any scientific term that I have explained, for example, I have explained symmetry here, right? What is symmetry? Symmetry is the overall shape of an organism, which means any organism that can be divided, the shape of the animal can be divided on one plane into two identical halves. This animal will be symmetrical. But if I put A to this term, that is symmetry or symmetrical, it will come asymmetrical. Always remember that whenever an A comes with a scientific term, it shows that the phenomena that we have talked about, the term, is absent in this type of animal or type of whatever we are talking about, right? Asymmetrical means that animals that have no plane of symmetry, they cannot be divided into two identical halves. And the example of asymmetrical animals are porifera, are poriferans or sponges, commonly called as sponges. Then there are two furthermore types of symmetry, that is radial symmetry and bilateral symmetry. Radial symmetry found in animals that have one main axis, and we can divide them into two identical halves by cutting it from any plane, but the plane should passes through that main axis of that animal. Then we can call that animal a radially symmetrical animal. And the example of radially symmetrical animals are cnidarians and echinoderms only. So here we have three phylums covered in asymmetrical and radial symmetrical animals, that is sponges, cnidarians, echinoderms, right? So how many phylums do we have in except these three? Always remember we have 9 major phylum. Out of 3 we have discussed under asymmetrical, radially symmetrical, which means rest of the phylum will come under bilateral symmetry. And what are bilaterally symmetrical animals? Animals that can be divided into identical right and left halves only by a single cut through the midline of its body. Any animal which can be cut through the middle line of its body into exact copy in the right half and exact copy into the left half, those animals will all will come under the bilateral symmetry, right? And the example of bilateral symmetry includes platyhelminths, nemathelminths, annelids, mollusks, arthropods, and chordates. Now the body cavity or coelom, coelom means body cavity. All the bilaterally symmetrical, as we discussed asymmetrical, radially symmetrical, we are not talking about them here. We are just taking the bilaterally symmetrical animals here. They can be divided into 3 groups on the basis of the body cavity or the coelom. These 3 groups are acoelomata, pseudocoelomata, and coelomata. This is the order according to the phylums, right? These are less or advanced animals, these are further modified animals, and these are the highly modified animals, comes under coelomate. But to remember these terms, I will suggest that you should study the coelomate first, then pseudocoelomates, and then acoelomates. It will be easy for you to learn. Coelomate or coelomata will include animals that develop a true coelom. And what is a true coelom? Body cavity or coelom formed by the mesodermal layer will called as true coelom, right? The body cavity present in the animal should be formed by the mesodermal layer of the germinal layer. Then only can call as true coelom, right? In coelomates, mesoderm splits open to contain a space that expands and forms body cavity. And this body cavity will have digestive, reproductive, and other organs develop right here in this cavity, and they will be suspended in this cavity when the adult is formed, right? The example of the coelomates are all the highly modified phylums that are annelids, mollusks, arthropods, and chordates. Now, pseudocoelomates, pseudo means false. The word pseudo means false. When it comes with the coelomata, that means it have a false coelom or the body cavity. How that can be? Simple. Animals which develops a body cavity, they have a body cavity, but they lack a true coelom, which means they have a body cavity, but that body cavity is not formed by mesoderm. So they are called as pseudocoelomates or pseudocoelomata. Example is nematodes. All the roundworms, nematodes are included in pseudocoelomates. As I told you earlier that whenever the A is added to any term, it means this phenomena is absent in this term, right? Acoelomate animals which lack a body cavity are called as acoelomate, and the example is platyhelminths. Fate of blastopore. What was the blastopore? We have discussed in the previous slides. Blastopore was the opening. While the development of the cells, the opening is formed when the three germinal layers are formed. So blastopore is an opening formed during gastrolith stage of embryological development. This opening forms either the mouth or anus of the animal, right? Either it will form either mouth or anus, the excretory part of the animal. Anus is the excretory part of the animal. Coelomates, all the coelomates only, not pseudocoelomates nor the acoelomates. Only coelomates can be divided into two groups according to the fate of the blastopore. That means either the coelomate will develop mouth from the blastopore or it will develop anus from the blastopore. On the basis of this, the animals are classified into two groups, that is protostomes and deuterostomes. If we break this word, proto means first, and stomes came from this word stoma. Stoma means mouth. This is very simple. In this group, blastopore becomes the mouth in adult. First, it means the blastopore will become mouth. Example, annelids, mollusks, and arthropods. Now the deuterostomes, the word deutero means second, and again stoma means mouth. In this group, blastopore develops into anus, right? And a second opening which develops later to form mouth, right? And the example of deuterostome is echinodermates and chordates.